Okay, here is part 3 of chapter 12 and 13, evolution. Let's talk about natural selection and how it shapes populations. There are a couple different ways that we've identified. There's directional selection. So let's take these two moths, and depending on where they live, what's their habitat, perhaps one phenotype is favored over another. So here's the two color morphs of a moth, and for some reason something happens and this one has a better chance to survive. We will see the curve move in this direction. That's called directional selection. Now, what if there's three phenotypes, and some animals have got multiple morphs, different colors, etc. And this is the original habitat, and this one is the most common because it has the best chance of survival. These are the outliers. But we see a split or a change in the habitat, and now there are new areas opened up, and so in one area these have a selective advantage, in the other area these have a selective advantage, and there really isn't a place where this guy has the selective advantage any longer. This is called disruptive selection. Even humans, okay? Stabilizing selection. This is when the intermediate phenotype is favored over the extreme phenotypes. And this has the number of individuals at the size, okay? Too small, too large. If you're too large, it can make for a difficult birth and can cause mortality. If you're too small and weak, you can also have mortality. Now, we have, through selection, just based on which babies survive, and medical technology has helped that a little bit, this is the optimal size, and so we see the majority of the babies are born in this range. Without medical technology, I think you would see the number of small and large babies decrease even more. Heterozygote advantage. Now, there is African sleeping sickness, and there is a genetic condition called sickle cell anemia. Sickle cell changes the shape of red blood cells and reduces the ability of the blood cells to carry oxygen. Okay? However, if you're normal and you don't have any alleles for sickle cell anemia, you're very possibly likely to get African sleeping sickness. It's spread by an organism called the tsetse fly, and it can be fatal. It kills quite a few people. Now, if you're a carrier for the sickle cell, that means you don't have sickle cell anemia because it's a recessive trait, and you would have to have two recessive alleles to display it. Now, two recessive alleles in sickle cell anemia is often fatal before reproductive age. But if you just carry one of the sickle cell alleles, but the other one's normal, you have a natural resistance to African sleeping sickness. You can tell I'm getting tired here, so sorry about that. And so that's the heterozygote advantage. Another example, heterozygote advantage, would just be your mutts. If you have a pet dog that's purebred, they have been so line bred that they have just basic flaws, physical flaws, more susceptible to disease, things like that. You go just pick up a mutt, and they're some of the healthiest dogs in the world because they are not homozygous in as many traits. Sexual selection directly influences reproductive success. So let's take these guys. These are bighorn sheep, and the males ram each other in the head, and the strongest one will run off the weakest one, and then the strongest one is the one that is allowed to breed and reproduce offspring, thus ensuring that his genes get passed to the next generation. That's one example of sexual selection. The females will pick the strongest most fit male. There's other things. Here's a bird of paradise. Many birds have got little ritual dances or maneuvers or courtship behaviors. Like this is a male that's this is an orange weaver and it's building a nest. And these behaviors attract mates. Female selection pressure or sexual selection is very important in just about every species of vertebrate. Females generally hold the power in most of these cases. It's the females that decide. Now, some other ways. Mutations, genetic drift, and migration. Mutations occur when, in the process of reproducing during meiosis, base pairs, basically nucleotides, get misplaced. They're not exactly 100% the copy of the original cell. Little errors occur, and so things change. And if that egg or sperm is fertilized with the mutations, it will now become a new allele in the population's gene pool. Genetic drift is another way. This is just by chance. The smaller the population, the larger the chance of genetic drift. And basically in a small population, for instance, if you take a group of animals into captivity to breed them because they're about to go extinct, they have a gene pool. And over time, through breeding, just randomly, occasionally, you'll lose some genetic diversity. We almost wiped out all the American bison in North America in the 1800s. And Theodore Roosevelt, one of our presidents, who was a naturalist and a sportsman, he wanted to make sure these animals did not go extinct. And so he formed some of the first national parks and natural areas and things like that. And he's the president that set up Yellowstone National Park to preserve the bison. And he did a good job, but unfortunately, we got down to less than 100 bison. So we lost a lot of genetic variability. And so the species is still very susceptible to a really bad introduced disease from, say, an introduced or invasive species. So American bison, while there are large numbers now, are genetically extremely genetically similar. That's called a bottleneck. When you lose almost the majority of the population, but just enough to survive, bring the species back. So that's one way, genetic drift. Another way is founder effect. Let's say you take a small group of people and they isolate themselves from the rest of society and they only breed within their small group. And we can see this in this is a group of Mennonites. I believe they're Mennonites. And within their gene pool, when they originally founded their original little settlements, there was genes and alleles to produce polydactyly, meaning 6 fingers or 6 toes. And since they don't allow people from the outside to join their communities very easily, they have what is called, they become more inbred over time, and the number of children in these groups that have 6 fingers or are born with 6 fingers is increasing. Now that may not necessarily be bad, but that's just an example. When you take a small group and isolate it from the rest of the species, it can sometimes magnify alleles that produce things like 6 fingers or diseases or whatever. Now here's an example of an ecological bottleneck. There was a large population of cheetah, and they died down to very few, and to the point that they're close to extinction. They don't reproduce very fast. They're an interesting cat, but they're not the same type of cat as the other ones. For instance, cheetahs can't retract their claws like other cats can, so they're a more primitive cat, and they're close to extinction. They're almost so genetically identical, meaning they got down to so few numbers that they're almost like clones of each other. And they have actually done studies where they did skin grafts from different cheetahs. And what they found is, normally when you do a skin graft, your immune system will reject that new tissue because it realizes it's different enough that it doesn't belong to that organism. The number of skin grafts that they did in which were completely accepted by the organism was massive. Very few rejected these grafts, which showed just how genetically similar they were. And then later, when we started running genomes on these guys, they are, for all intents and purposes, very close to being clones of each other. Evolution occurs in some other ways. Migration. You can have two separate populations, and then sometimes animals, individuals, will migrate to other populations and begin breeding. And there you go. You get new alleles introduced into the gene pool. Here was an interesting case study. How do we increase the amount of fish harvested from the oceans for food? Okay. There's an Atlantic silverside. It is harvested commercially for food, and there are fishing regulations about it, and the regulations basically say that if fish, in order to take fish, they have to be a certain size. Okay? So they can take the larger fish, but they can't take the smaller ones. And here's what happened. When they were only harvesting the large fish, over the course of generations, what they saw was a drop in average size because they were taking the mature large fish and there were some mature smaller fish, and those were the ones that stayed behind and bred. So the number of small fish actually decreased or increased, and the larger fish were pulled from the gene pool. Okay? Now, when they just harvested the small fish, they saw an increase in the yield of the fisheries. And when they just harvested random fish, they didn't, throw anything back, they found an equilibrium that kind of bounced and oscillated between 3.5 and 4 on this scale. And so we can use science to determine things. So the laws require fishermen to do this when in fact that's probably not the best thing to do. Yet these regulations have not been altered. And fossil evidence. Let's look at fossil evidence. We have what is called a timeline of Earth. First fossils started showing up about 3.6 to 3.8 billion years ago, and these were all single-celled organisms. But fossils are just remains of past species. A transitional fossil is a common ancestor for two different groups of organisms. They allow us to trace the descent of organisms. And so as we find species of fossils, we fit them in to current lineages based on physical similarities. We're now figuring out ways to isolate DNA from fossils. We get little scraps and bits and pieces of it. And, maybe in another 100 years, we'll have some pretty close genomes. But we can now apply phylogenomics to it as well. Clues to evolution lie in these structures. We can compare them. And that's what paleontologists do. They dig up fossils and study the remains. Now, most people think of paleontologists as guys who go dig up dinosaur bones, but there are lots of other different types of paleontologists. There are paleontologists that study plants or mammals or whatever, okay, insects. And all of these are important to look at. There's other fossil types that we look at. Now, understand a fossil is just a replica. What happens when something falls into an area, gets covered over with silt very quickly and isolated from oxygen, it doesn't break down organically, and slowly over time, and it takes a long time to make a fossil, each individual molecule of the living organism gets replaced by a molecule of rock. And so it makes kind of a model. But sometimes the rock that forms over the fossil seals an inner core of organic material that is in an airproof pocket, and so it's still there. It never gets broken down, it never gets replaced, and we're finding ways now to grind up fossils and find these little scraps of DNA from the leftover organic material deep inside the fossil. Other types of fossils that we look at, here is basically coprolite. It's poop for all intents and purposes. And fecal matter like this, we can find it in association with other with organisms, and we can get an idea what did they eat. There's petrified wood. Here is a petrified egg. This is a fossil of an egg. Fish, plants, ammonites, or trilobites rather. This is a dinosaur. That is a Triceratops. And these fossil records is incomplete. Not every species that has ever lived probably came into contact and was fossilized. And then some species were fossilized and destroyed, or damaged, or found and not noted. And we're still finding new fossil species, but we'll never have them all. Okay, that's just something that we understand. We're never going to find them. But we can do things like take the model of our lineages and say, okay, if we have something between this and this, it should be in rocks this age. And then we go look in those rocks, and we find a species of fossil that has characteristics from the two on either side of it. Okay, and that's making predictions. Fossils record evolution. That's what these are sedimentary layers that are laid down over millions of years. And how sedimentation works in fossils, here at shorelines, things are buried by silt, and so they don't decompose rapidly enough, and they turn into fossils. Archaeopteryx, this is a transitional fossil. There are people that claim this is a fake. The problem is, this one, the first one, showed up in this quarry in Germany where they were quarrying rocks, and they're still finding them. The number of Archaeopteryx are increasing because they find them all the time. They're getting more common. And so this has got bird characteristics, it's got reptile dinosaur characteristics. It would be considered a transitional fossil. Okay? Basically, every species either becomes extinct or is a transitional fossil because it will evolve into another fossil or another species. For instance, Australopithecus afarensis and Homo sapiens, there's about 330,000 generations between these two species. And things are going to change in that many generations. Okay? We're seeing things change even today. Anatomical evidence. We can look at that. There are what are called homologous structures. And homologous structures basically have the same set of bones in similar ways. They're modified extensively to meet different adaptive needs. But you can take the flipper of a whale and the leg of an antelope, and they both have the same number of bones in the flipper and the leg. It's just they're shaped differently because the animals live in different environments. Okay? There are a couple things. All vertebrates have a tail and gills. Okay? Those are adaptations to live in an aquatic habitat. And we don't continue to have them because the genes or alleles that produce the enzymes to maintain these structures until we're born have been mutated. They've been altered. Okay? The alleles have been altered, so they're not there anymore. Here is some examples. You can look, whether it be, there's a hawk, a seal, a lion, a bat, a human arm. We all have the same bones. And they're all homologous. My right arm is homologous to the right leg of a lion, to the right flipper of a seal, to the right wing of a bird of prey, and to the right arm of a bat. Okay. Homologous structures They don't have to have the same function or look exactly alike, but since they all have the same number of bones, that is indicative of a common ancestry. Here's some more. This gives you kind of a better look at some of these different homologous structures. Now, there is a vestigial structure. This is a structure that has lost its function. It's homologous to functioning organs or structures on other species, but they're just pretty much no longer needed in the current species, but they still have the genetics there for them. Snakes, especially boas and pythons, have got little vestigial hind legs. And the males actually use them in courtship for the females. But they're not very functioning. Vestigial limbs in whales. This is one of the ancestors of whales. Whales actually evolved from a series of organisms that were primarily land animals that started spending more and more time in the oceans and the water and the shorelines. And at some point, they went from freshwater to saltwater. This is a link to a video. You can open that link to the video if you're interested in this, and there's a paleontologist where he works, and you can actually see how a paleontologist works, how they do what they do. It's pretty interesting. Now, analogous structures. These are different. These are structures that basically function the same or look the same, but they don't come from a common ancestor. They just come as response to organisms living in the exact same environments. So these little salamanders, there's a lot of species of blind salamanders that live in caves. But they're not all that closely related. They're related to a species that doesn't live in the cave that is almost conspecific, meaning living in the same streams or lakes or whatever next to the cave. And they kind of separated because of that. So this is Typhlomolgi rathbuni. That's the Texas blind salamander. You have one here in Tennessee called the Tennessee cave salamander. And those are related to salamanders in the vicinity of those two places, but they're not very closely related to each other, even though they both are blind and they're this color and they live in caves. The reason that they look like that is because they live in caves, not because they were genetically related. Convergent evolution, and that's what this is called, is basically same functional characteristics or physical appearances occur because they share the same environment or habitat. So there's some examples. Here's a good example. Emerald tree boas and chondropythons, or green tree pythons, depending on how you want to call them. This one's from South America. This one is found in New Guinea. And they don't share a direct common ancestor. They both evolved separately because they both adapted to similar environments. Summary. Homologous structures, basically they're similar because they're inherited from a common ancestor. Analogous structures serve the same function, but they don't share a common ancestor. Vestigial are reduced or obsolete function in similar groups. Embryonic development patterns provide evolutionary clues. There's a chimp and a human fetal skull, very similar, but at the adult, they're very different. In fact, baby chimps, a 3-year-old or a 2-year-old baby chimp is smarter than a 2 or 3-year-old human. But then they start falling behind as they move into adulthood. Some similarities between different embryos. This is a drawing. I need to get some pictures of this. And so I did. Okay. Fish, mouse, alligator, very similar. Embryonic development patterns. This has led to the development of developmental biology. And there's a video called Evo Devo, which is a very creative way of explaining developmental biology. And developmental evolution. But basically there are certain alleles that are very powerful and have a huge impact on physical appearance of organisms. These are called the HOX or homeotic genes. And so basically how they're coded determines whether you have a tail, whether you have legs, whether you don't have a tail, etc. These Hox genes have been mapped in a lot of different animals to compare them. All right, and that's what gave rise to developmental evolution, because in addition, we found, and it's kind of hard to explain, and I'm not the most knowledgeable on this. I'm still reading about it and learning about it. It'll take me several years to probably get a really commanding knowledge base on it. But alleles don't just function at one level. If an allele is turned on, it might affect 15 other alleles. And when it's turned off, it might affect a different 15 alleles. And so there are multi-levels of these alleles turning off and on that affect our physical characteristics. DNA and biochemical evidence. Almost all living organisms, we use the same biochemical molecules. We use DNA triplet. All of our DNA is basically the same. There aren't different amino acids in this animal that don't exist in this animal, etc. Here's just the significance. We can look at this one protein and the coding for it, and basically we can count the number of differences, okay, in just this one set of DNA. And so we can then set up these cladograms or phylogenomic sequences to show what we are most close to based on the similarities. So if we've only got 35 differences with a monkey, and maybe 50 differences with a pig, and 150 with a duck, and et cetera, et cetera, et cetera. And this forms the basis of genetic determination of evolutionary relationships. Comparing the DNA and protein sequences determines evolutionary relationships in incredible detail. Okay, there it is, molecules, relatedness. Evolving backwards. I mean, sometimes we evolve structures and then we lose structures. Snakes evolved from lizards. Okay, snakes without limbs burrow more easily than those with limbs. Cave salamander. This salamander doesn't have eyes anymore. Now, eyes are a big evolutionary improvement, but this animal chose to inhabit an area where it really no longer needs eyes. There's no light in there, so eyes don't help it. So why keep wasting resources building eyes if you don't need them? Evolution of the eye, we have multiple organisms that have intermediate steps towards what we have, an optical eye. In essence, evidence is tested by the scientists, and we try to prove that it's false. And we can't. And that's when we accept it. All right? Now, you can choose not to accept the evidence for a scientific discovery, and that is certainly your right. And who knows, maybe you're right, maybe it is false, because science will never know everything 100%. And as a scientist, I have to accept the possibility that I might be wrong. And so I always, when I do an experiment, I'm always trying to disprove something. I'm never trying to prove something. And if I can't disprove it, then I know that evidence is valid and it supports the hypothesis. And that's what evolution is. Evolution is supported by tremendous amounts of evidence. A lot of people don't understand it because it takes a lot of scientific literacy to understand every aspect of the evidence that supports evolution. Even I can't ever in my lifetime absorb all the knowledge about all the evidence that supports evolution. And so that's why we specialize in certain areas. Evolutionary biologists are about the closest, but they're not as common as some other types of scientists. The evidence is there. You know, you have a right to choose to accept it or not. However, whether you choose to accept it or not, the evidence is still there and it has never been disproven. Because every aspect of evolution comes from testable hypotheses. we can run experiments to test and see if we're wrong, because we want to prove that we're wrong. Scientists are the biggest skeptics in the world. And that is the wrap up for this topic. Thanks.